Hello students, today in this session we will discuss about the gene regulation or regulation of gene expression. Regulation of gene expression. Okay, first of all we have to understand in a living cell, okay, many metabolic activities will take place. And we know that metabolism includes both anabolism and catabolism. Means what? Anabolic reactions and catabolic reactions, both together called as metabolic reactions. Of course, all the reactions are biochemical reactions, are catalyzed by enzymes which are mostly proteins. So these proteins are formed only after translation with the help of mRNA, and mRNA is formed only after transcription from the DNA. Okay, and so we have to understand that many genes are there with the DNA present in a living cell. And will all these genes are expressed at a time? No, not at all. We know that if all of them are expressed at a time, so then it becomes, there will be a traffic jam. Okay, but actually without any mishap, this traffic is going on in the cell. And remember, some of the genes are regularly transcribed, and as a result of which proteins are formed, and as a result of which that product is formed, because these enzymes, when they catalyze those reactions, the products are formed. And such genes which are regularly transcribed, which are required, okay, are called constitutional genes. Called constitutional genes or housekeeping genes. Constitutional genes or housekeeping genes. What are the examples we can give for such constitutional or housekeeping genes? Of course, we know that do we respire only during daytime? Or do we respire only in the night time? Or do we respire only at a particular time? No, we respire all the time. And we know that we respire aerobically, and the aerobic respiration involves glycolysis, okay, pyruvate metabolism or link reaction, and that TCA cycle or Krebs cycle, and also electron transport chain or oxidative phosphorylation. And all these reactions are catalyzed by different types of enzymes. And the information for these enzymes is with the DNA. So those genes which are required for respiration, or which are required for glycolysis, which are required for that link reaction, which are required for that Krebs cycle, they're all regularly synthesized, and because of which regularly these enzymes are produced in the cell, and that's why regularly our respiratory activity is taking place. So that is the best example for what constitutional genes. But certain genes are transcribed only when they are required. Okay, so and all those genes which are transcribed only when there is necessity or only when they are required, they are called non-constitutional genes or luxury genes. Non-constitutional genes are luxury genes. So then how their expression is regulated? So that is what gene regulation. So their expression is regulated. Okay, whenever they are required, so they are expressed, and whenever they are not required, their expression is stopped. That is what is gene expression regulation, or regulation of gene expression. And of course, what is the example we can mention here for in plants? In plants, of course, you see, the gene responsible for the production of nitrate reductase. So that nitrate reductase is required only when there is availability of nitrates. Okay, so when nitrate is not available, so then there is no need of that enzyme. So that's why it is regulated. That gene for nitrate reductase is regulated. Like that, we can quote so many examples. And okay, and remember, like that, you just remember that in a cell, we will see two kinds of genes: constitutional genes or housekeeping genes, and which are okay, always expressed. And non-constitutional genes or luxury genes are expressed only when they are required. Okay. And of course, remember this regulation of gene expression, it occurs at what time? Okay, it occurs right from the beginning. Okay, as soon as the zygote is formed and the zygote is becoming an embryo, and embryo is developing into an organism, you will find what so many sets of genes will be regulated. That's what I wrote here. Development and differentiation of embryo into adult organism is the result of coordinated regulation of several set of genes. So several set of genes are okay, regulated. That's why at some places some different kinds of organs are formed. Actually, don't forget, we all started our life with a single cell, and that single cell is zygote. And the zygote first divided mitotically and developed into embryo. And that embryo now is undergoing what differentiation. At some places, some cells are differentiated into arms, and some are differentiated into okay, legs, and some are differentiated into head, and some are differentiated into brain. And you see, that means what actually all the cells which are there in our body are originated from the single cell, that is what zygote. But this differentiation is due to what the coordinated regulation of genes. So that's what you had to understand. And with that, this expression of gene is under control. The expression of this gene under control of what metabolism, because the metabolic rate of an organism is not always same. And even if we take our example, our metabolism rate is not same even in one day. In the morning it may be more, or in the afternoon it may be more, or in the night the metabolic rate may be less. So it depends on it is under control of that metabolic rate, and it depends on the physiology. Means what is the functional ability of that organ, when that organ has to work fast. So the genes which are required for that will be okay, expressed. Like that, even environmental conditions may also control this gene expression. Because always throughout the year, our body will not have the same condition, or we will not have the same feelings throughout the year. Okay, that depends on the environmental conditions. That all due to what this gene expression which is under regulation. Okay, that's why this regulation of gene expression is a very broad aspect, and we can't discuss all aspects here, but let us discuss one aspect. If you consider, suppose, if we consider the gene expression resulting in the formation of a polypeptide chain or protein, suppose if we take this condition, means what, the gene is expressed and a polypeptide chain is formed. That is what, during translation, the mRNA is translated into a protein. At that time itself, the gene is regulated at several levels. At that time also, during polypeptide formation also, or during protein formation also, it is regulated at several levels. So, for example, in eukaryotes, in eukaryotes, okay, at the time of this protein formation, of course, the gene is regulated at four levels. One is transcriptional level. One is transcriptional level. And the second one is processing level. The third is transport level. And the fourth one is translational level. Okay, and remember, in eukaryotes, this transcription is not like that of prokaryotes. Okay, now what is the difference? In prokaryotes, as a result of transcription, mRNA is formed. But in eukaryotes, okay, in eukaryotes, as a result of transcription, hnRNA is formed. What is this hnRNA? Heterogeneous nuclear RNA. Heterogeneous nuclear RNA. So it is composed of exons and introns. Exons are coded information or coding information, and introns are non-coding information. Okay, and the presence of exons and introns is called split gene arrangement. Presence of exons and introns is called what? Split gene arrangement. And this hnRNA cannot be translated because it has so much of useless information. So that's why the second level is what? Processing level. So you can call this hnRNA as a primary transcript. Primary transcript. And this primary transcript should be corrected. And that is what is called the processing level. And during processing level, so these introns are removed and exons are jointed. And this removal of introns and joining of exons is called splicing. Removal of introns and joining of exons is called what? Splicing. As a result of splicing, what is formed? Now you get what? mRNA, messenger RNA. And remember that during this processing, almost 50 to 90% of HNRNA or primary transcript is discarded. Okay, 50 to 90% is discarded. That means, you see, what is the amount of intronic information? Intronic information amount is 50 to 90%. So exonic information is 10 to 50%. Okay, so that much is removed, and then only we get this mRNA. And don't forget that this mRNA is formed inside the nucleus in eukaryotes, and for translation, it should come out of the nucleus and should enter into the cytoplasm. And how it happens? And it can't diffuse through the nuclear membrane. That's why it will search for the nuclear pores, and through nuclear pores, it will come out and enter into the cytoplasm. That is what transport level. And after reaching this cytoplasm, so this mRNA should get attached to the ribosome. Of course, remember that ribosomes will have two subunits, one smaller subunit, one larger subunit. And remember, even the environmental conditions or physiological conditions also regulate the gene expression. And there is no guarantee that this mRNA, which has entered into the cytoplasm, will be definitely translated. And remember that magnesium concentration is required there. And remember that the smaller subunit, when it gets attached to the mRNA, and now the first initiation tRNA will attach, and to this complex, the larger subunit should be attached, and that happens only when there is optimum concentration of magnesium in the cells. So don't forget, for binding the two subunits of ribosome, optimum concentration of magnesium is required. Okay, I hope you are understanding. Under low concentration of magnesium, they will not bind to each other. So that's what there also, during translation level also, it is regulated. Like that, in eukaryotes, so the gene expression for the formation of a protein is regulated at how many levels? 4 levels. And when we come to the prokaryotes, so this rate of transcriptional initiation is the predominant site for control of gene expression. Means what? At transcription level only, so the gene expression is controlled in prokaryotes. Okay, so in prokaryotes, okay, the rate of transcriptional initiation is the predominant site for control of gene expression. So that means what? At transcription level only, the gene expression is regulated. This is because in transcriptional unit, you know that the transcriptional unit will have 3 parts. One is promoter site, and here you will find terminator site, and this is what? Structural gene. Structural gene. And now the promoter site will be occupied by the enzyme RNA polymerase. Okay, if this RNA polymerase activity okay, is regulated by interaction with accessory proteins. So the RNA polymerase activity is regulated by the interaction of accessory proteins, which are also called as regulatory proteins. Regulatory proteins. And these accessory proteins or regulatory proteins may behave as activators. Then the regulation is called positive regulation. And sometimes they behave as repressors. Then it becomes a negative regulation. Okay. So now that is what a transcription level. RNA polymerase, when it wants to transcribe transcribe this mRNA. So whether it is going to transcribe it or not will depend on these accessory proteins. Okay, are regulatory proteins, and if they activate, then it becomes positive regulation, and if they suppress or which are called repressors, then it is called negative regulation. That we can understand with a concept called operon concept. Okay, that we will discuss now. Okay, let us try to understand this operon concept. And this operon concept is a coordinated group of genes. It is coordinated group of genes. It is not one gene. It is coordinated group of genes, and the group includes a structural gene, operator gene, promoter gene, and regulator gene. A structural gene, operator gene, promoter gene, and regulator gene. And all of them function together and regulate a metabolic pathway. So all these genes function together and regulate a metabolic pathway. Okay, and for example, lac operon, okay, TRP operon, ARA operon, his operon, valve operon. Of course, this is for the metabolism of lactose. This is for the metabolism of tryptophan. This is for the metabolism of arabinose. And this is for the metabolism of histidine. And this is for the metabolism of valine. Like that, these are few examples, and you will find many such examples for the operon. And this operon concept was given by Francois Jacob, a geneticist, and Jacques Monod, a biochemist. Francois Jacob, a geneticist, and Jacques Monod, a biochemist. Of course, in our syllabus, we have one operon, that is lac operon, and we will discuss that lac operon in detail in the next video.